Welcome everybody for this second session on the data track. Unfortunately, we missed one of the speakers, the third one, Luis Rueda, because apparently he's ill and he couldn't come. So we will have only two talks today, the two proceedings. And I'll remind you that there is another session later in the afternoon and another session tomorrow, so if you can keep on this track. So my name is Josep Lluís Gelpí from the Barcelona Supercomputing Center and the University of Barcelona. I am the chair of this thing, apparently, and also from Elixir Spain. And for me, it's a pleasure to have this room so full. So keep going, keep going. There's plenty of space around. Don't be shy. I tell this to my students. Nobody won't come to the first row. Okay, so no more delay. We're on track. So our first speaker today, if I don't mess this around, is Alexander Dietrich. Is that correct? That, as it says there, is going to talk about the bias-aware simulation of bulk RNA-seq data with variable cell type composition. So 15 minutes. Thank you.
Okay, thank you everybody. Thanks for attending. So I'm a PhD student from Munich, from Technical University of Munich at the Chair of Experimental Bioinformatics. And I want to present my project here, which is SIMBU, a bias-aware simulation of bulk RNA-seq data with variable cell type composition. Okay, so like when we look at tumors, they are usually formed as this aggregation of different kind of cell types. So we have stromal cells, malignant cells, of course immune cells as well. And like in the recent years, and with getting to know tumors more in depth, this so-called tumor microenvironment has played a more essential role in the control and study of tumors. You can see this on the right side, the figure where we have like different subtypes of the T cell, and they have like different prognosis effects on the tumor. Some of them, like many studies find a positive effect, and for others, many studies find a negative effect. So this just goes to show how important this tumor microenvironment is in cancer therapies. But not only in cancer, this is one important thing. Also in neurodegenerative diseases, the immune cells are quite an important role. Also, medication can impact the immune cell environment. And also in a more like classical bioinformatic approach, when you look at differential gene analysis, the cell type composition is also one well-known confounder. So what we can do to study these cell type fractions experimentally would be like very well-known and well-established methods like FACS, what you can see on the left, where you can sort like for specific cell types using markers, or immunohistochemistry, or here in the middle, where you can stain for specific cell types in a tissue sample. Right, so since these are like experimental assays, they have a few drawbacks. First of all, they are quite limited in the number of cell types that you can study simultaneously. Also, if you like thinking about adding these methods into your like experimental workflow, they like add additional steps of labor and cost, of course. And also, these different cell types are quite biased in the capture efficiency, which also is another hurdle. So, since we are here at the bioinformatics congress, we also have in silico approaches to study this cell type composition, which is then called cell type deconvolution, where the goal is basically that you look at the bulk RNA-seq sample that you have. And basically, you want to know what amount of cell type fractions are in this sample. And what these methods, these cell type deconvolution methods, then do, they create these signature matrices where they have, for each of the cell types, the different marker genes set up. And together with the input bulk data sample, they can then infer the cell type fractions. Okay, now the issue is we have different methods available for the cell type deconvolution. But I mean, this is a computational approach, so in a way we have to trust these methods that they do their job correctly. So, but we are not 100% sure if they really estimate the very correct cell type fractions. And to account for this, or to like benchmark these deconvolution methods, you could then either go the experimental route again, where you say, okay, I will just then do my FUX analysis of the Spark sample, and then I could just compare what the FUX put out for the cell types and what I put out with my deconvolution method. But this is like, in a way, this lacks a lot of flexibility and also scalability, since you're like, as I mentioned, you have all these drawbacks for these experimental methods. So this is why we proposed Simbu. So this Simbu is a software package which allows you to create artificial bulk RNA-seq datasets, and where you know the cell type fraction. So this is the important point here. And therefore, you create a kind of gold standard dataset that you can use then to benchmark different deconvolution methods. Right. So now we'll go a little bit into detail how this works, how the method works. So what we are doing is we use single cell datasets, so where we have an annotation, a cell type annotation for each of these single cells. For example, here we have like 3 different cell types. And what we then do is we sample these single cells in a known fashion, and this can then gives us our simulated cell fraction. So we'll say, okay, cell type 1, we want like 20%, cell type 2, we want 30%, and so on. And then we use all of these expression profiles of the single cell and create this pseudobulk profile. So we have multiple cells in one pseudobulk profile. For Simbu, there are different ways of getting these single cell expression profiles. We added the option to download them from the Spira database, which has over 150 annotated single cell datasets. But of course, you can also use like any other annotated single cell dataset here as well. Okay, and yeah, what you can do with this pseudobulk profile then, of course, as I mentioned, you run your deconvolution method on this profile, you get some estimated cell fractions out of this method, and then you're able to compare between these two, the estimated and the simulated fractions, and get some kind of validation of the method. Okay, so now before I go into a little bit more technical detail and the results of our method, I want to introduce one important bias that has to be taken care of when simulating these pseudobulk profiles, and this is the amount of mRNA between cell types. So what you can see here are 3 different single cell data sets, and the columns basically are then the different cell types. And these basically here show that the total mRNA so can differ between cell types. So for example, what we can see here, this green and the brown ones, these are B cells and dendritic cells, and like we argue that the total amount of mRNA like reflects how the cell types also differ in their number of expressed genes, or also in the number of mapped reads to this cell type. And this is one bias that has to be taken care of. So the mRNA is different between the cell types, and this is what we are trying in Simbu. So what we are doing, like as I mentioned before, we pick these single cells here. We have our single cell matrix. We sample these single cells, but what we do previously or in between there is we introduce scaling factors. So that means we basically weigh the expression profiles of the single cells according to a mRNA bias that we calculate, or a scaling factor that we calculate. So in Simbu we have different ways of calculating these scaling factors. We have data-driven experimental derived approaches. For the experimental approaches, these are from publications where they used like experimental assays to measure the amount of mRNA in the cell type. And the data-driven approaches use then use single-cell profiles to estimate, so to say, the mRNA levels in the cell types. And yeah, these scaling factors are then introduced between the sampling step, and then the pseudobulk profile basically has this bias between the cell types integrated. Right, and what we also then tried is we wanted to look at how these scaling factors compare with each other. So we calculated the correlation coefficients between all of our selected scaling factors. So what we did here is we had our experimental scaling factors down here, And then we had 3 different single-cell data sets, and for each of them we calculated 3 different data-driven scaling factors. So we calculated like the number of expressed genes per cell, the number of mapped reads per cell, and also the algorithm called Census, which also tries to estimate the lysate transcript counts in the cells. And what we found here is then that the number of expressed genes, they correlate quite well with the so-called gold standard scaling factors, which we would argue are these experimentally driven assays, as they have like experimental evidence for the value or the amount of mRNA. So this is one very nice result, basically, because that means that we can use just a very simple approach, just count the number of expressed genes per cell, and this gives you a quite decent estimation for the mRNA in this specific cell. So what we did then in the end finally was we used these biases, of course, and first what we wanted to look at is how an extreme bias would affect the deconvolution. So what you can see here is we ran one deconvolution method called QuantiSec, and we simulated again different pseudobulk profiles, and then we compared how this method like estimated these the cell type fractions. And the catch here is that we, like for each of these boxes here, for one specific cell type, we like added a very extreme bias. So we wanted to see how this bias affects then the results in the deconvolution. And what we can see, and what is also expected, is that if we increase the bias, like the mRNA bias for the B cells here, our deconvolution tool drastically overestimates then the cell type fractions of the B cells. And this is also present for other cell types, and also not so much for like monocytes in this case. But it still shows that the bias is something that really offsets the deconvolution tools if it's extreme. But of course, this is only like a proof of concept. And what is more interesting is when we look at like the real world bias, so like the biases that I was introducing previously. So what we did here is basically we tried all of our data-driven scaling factors, introduced them to the deconvolution methods, to the pseudobulk profiles, and then ran the deconvolution again. And with QuantiSec, this is one deconvolution method which can handle this bias. So they are a bias-aware deconvolution method, meaning that like they rescale their results in the end. And what QuantiSec can do is it can be run with and without the scaling. And what we can see here on the top is that if we do not do the scaling in QuantiSec, it again overestimates certain cell types here. These are the macrophages again here. But then if we like run QuantiSec in its like default way, so how it's supposed to be run, this bias is then more related in a way. So what does this mean in the end? I mean, this Simbu, the method, is basically a tool to benchmark deconvolution methods. And what we've found out here is that it's very important, if you want to benchmark these methods, that you also integrate this bias, because then you wouldn't have this comparison to see that this deconvolution tool you're looking at can take care of this cell type bias. Okay, and so now I just want to reiterate again what you can do with Simbu and how it can be useful for you. So as I mentioned, you can use it to simulate different pseudobulk profiles, of course with known cell type fractions. We also have like different ways of generating these cell type fractions. You can like create them customly, however you like to have your profile, but also we have like a random scenario where we have all different cell types with random fractions in a way. Or also, what you can also try out is that you only use one single cell type in your pseudobulk profile and then see how the deconvolution tools react to that. So it's quite versatile in that way. We also have a very easy access to multiple annotated single cell data sets. Also, what's cool here is that these are from several tissues and also from organisms and sequencing methods. So that means you can also look at how the deconvolution tools work on different tissues as well. Yeah, and then we argue that this mRNA bias that you can integrate in the simulations is very important to like better represent these cell type specific differences and also benchmark the deconvolution tools in the end. And yeah, finally, we think that SIMBU can not only be used for classical RNA-seq deconvolution. You could also, of course, I mean, it always depends on the amount of input data. It doesn't have to be single-cell RNA-seq. You can also go, use single-cell DNA methylation data and generate the DNA methylation profiles, and the same goes, you can, like, use cases go to spatial transcriptomics, also a TextSeq. So, this brings me to the end of my presentation. SIMBU is available as a R package. It's also was accepted to the bioconductor environment and will be released there in the next release run. And yeah, I want to thank all of the people who were involved in the project. And yeah, this is our group. You can check it out. And the preprint is also available and now also online as a proceedings paper in bioinformatics. Thank you.
Plenty of people. So thank you for adjusting to the time. That was perfect. Very nice talk. And we are plenty of time to have questions. Mm-hmm.
Hello, a very nice talk. Just maybe a general question from my side. You know, single cell is something that, to my view, is getting quite mature, for example, to be applied also in industry and pharma companies, for example. And this package, SIMBU, might be something that is of very interest to invest, for example, now and in the future. I was wondering what's your strategy, or if you have a strategy in mind about, for example, long-term support of a package like this, or industrialization and, you know, support and updates in the long term. Do you have any strategy about that, or any comment on this?
So not a detailed strategy plan, of course, but I mean, since it's in the bioconductor environment, it's in a way in a very polished state already, with all the checks that go through there. Of course, I mean, it's mostly that I will handle the updates and bug reports and everything, and I guess this is then, always comes to me, and I'm really happy to integrate all the updates and everything that will come on, like new ideas that we can have to the simulator. So yeah.
Thanks a lot.
Thank you very much for your talk. So I just have a question regarding the scaling factor and the idea behind it. So my question is, with single cell, you could use CPM, for instance, and then you would basically correct for the scaling factor that you have in cells. Why don't you use CPM for the simulation? What's the reasoning behind having this external factor? Is it because you want raw counts?
So we can do the simulation with raw counts and CPM or TPM, or whatever you have, basically. But we say that this scaling factor is one additional bias that has to be added after the normalization again. Does it make sense again?
Okay, maybe I need to think about it more, but my thought was just, if you correct for the scaling factor before you mix it, you could have the equivalent of, I don't know, FPKM basically in your simulation, and I don't see why we would have to re-add it, but maybe we can discuss, so we can discuss this. Yeah. Thank you.
Thank you for your talk. So in addition to the total mRNA content differences across cell types, we have also observed that there are differences in the ribosomal read content that can go from 5% to 60% in some cell types.
Okay.
Have you addressed this bias, or do you think this could also be?
No, we didn't. But that's a good point. I mean, that's then comes back to the guy in front, where we say, yeah, we want some more progress and more updates to that, and that could be also one scaling factor that we could combine or have two separate ways of adding them. But yeah, not implemented now, but hopefully soon.
Some years ago, a reviewer made us create an experimental ground truth for cell type deconvolution algorithms, which was instead of simulating, we FACS sorted different proportions and then performed bulk RNA sequencing. Did you ever benchmark your deconvolution methods against something like that, like an experimental ground truth?
Yeah, of course. I mean, of course, we also have bulk data sets with experimental, with FACS data sets. So the question is how this simulated data sets compare to the real data sets? Or?
Versus a real experimental ground truth.
So yeah, I mean, we didn't do this in this project, so this was more about the simulator itself, so not about the deconvolution, a benchmark. But I mean, in a benchmark that I think that they usually do the experimental side, so bulk samples with fax data and also simulations as well. Yeah, both. But it's we didn't do that here.
I have a quick question regarding to this. Can you do that the opposite way? Can you use that for kind of quality control of the experiment?
For what, sorry?
To control the quality of the experimental results, to detect that some cell is wrong or something.
I guess, but
I'm informaticians, not experimenters.
So I would trust the experimental values more than I would trust the deconvolution results of a simulated sample. But I think the thing here isn't that you want to like compare these these two deconvolution results, but it's more like that you have more flexibility in how you can set up the deconvolution benchmark. Like with FACS, it's quite limited. It's a lot of work additionally. And when you say, oh, let's try this, like different tissue maybe, different cancer type, how it performs there, then you have to basically start over again. And here you basically have to only find a annotated single cell data set from the tissue, and then you can go ahead with it.
No questions. We miss one talk, so we can do a lot of questions. No, I had another one, but because I'm not an expert, probably it's not a nonsense. But can you do this with your own data? I mean, do you need a lot of data as a reference, or just my experiment I can use for
So you mean like as an input data, right? For a single cell?
So
Yeah.
A normal experiment in a normal situation, is that enough?
Yes, that's enough. I mean, the thing is, it always depends how many cell types you have in your single cell data set. The cell types you have in your single cell data set, those can be simulated, of course. And I mean, as these cells are sampled with replacement, of course you need a few of each cell type, but I would say that like 100 of each cell type would be a good starting ground. Yeah.
Okay. So I think there's no more questions, so thank you very much.
Thanks.
So next speaker is Jacob. I don't dare to pronounce your surname. Slides down.
So, yes. Hi, my name is Jakub Bartoszewicz. I have just recently finished my PhD studies at the RENA lab at the Hasse-Plattner Institute. And I'd like to show you our work on detecting DNA of novel fungal pathogens with data and with deep learning. And this is joint work with Therdis, who's also here, and also with Melania. So unfortunately, we all could kind of feel that novel pathogens are a threat and a lot of concern, but it kept happening, it keeps happening, and it will keep happening, unfortunately. So novel pathogens will keep emerging because they evolve very quickly, and with the climate change, there are some reasons to believe it's just going to get worse. Here I'm going to focus on natural pathogens, but some of those methods, or most of them actually, could be also used for synthetic pathogens if they're ever materialized. So we hope we could find them quicker. Not sure what that cursor is, but okay. If you try to find a pathogen right now, you would probably use a targeted test like a PCR or like the antigen test, but if you don't know what you're looking for, and that would be the case with novel pathogens, you would have to use an open view perspective. And the idea is that you would sequence DNA in your sample And then you would need some computational analysis to understand what's going on. And of course, the standard would be to use standard bioinformatics approaches. So either alignment or K-mer-based approaches basically match your reads or your context to a database of known pathogens and non-pathogens. The problem is that if your pathogen is really novel, you may not have the correct reference, and you will just miss all those reads or all those contexts, and you're going to be left in the dark. For that, we have been developing deep learning solutions that can analyze reads or contexts and assign them to pathogen classes, even if the reference is unavailable, and that usually works a bit better for those novel pathogens than alignment-based approaches, even BLAST. And then you can maybe filter out some interesting reads and have some follow-up analysis on that. But we have nice methods for viruses, we have nice methods for bacteria, and those are those big famous classes of pathogens, but fungi are really important as well. And we noticed that whether we wanted to use sequence alignment or deep learning, actually databases that would help us with that were maybe not fully missing, but slightly incomplete. So why fungi, you could ask? Well, they are actually pretty important. So for a lot of population, they're pretty benign, but in susceptible populations, you can get very high mortalities, like up to 95% in some cases, which is just scary to me. And you also have novel species being discovered and emerging, like Candida auris, which was discovered like not even 15 years ago, and is already one of the most important drug resistance threats all over the world. The problem is we would like a database that would be fair, so findable, accessible, interoperable, and reusable, but would also be manually curated and comprehensive. And there are a lot of data resources related to fungi, but we haven't found one that would satisfy all those points, at least not to the extent that would really allow us to reliably detect novel pathogens. So we had to create one. And in this talk, I will show you those 3 steps. We first wanted to create this database that would help us in novel pathogen identification for fungi. Then I will show you what we did with fungal pathogen classification, that is detecting if a novel fungal species is a pathogen of humans or other non-human organisms, and also a bit about visualization of our database. And at the end, I will talk about multi-class classifiers that you could actually use in metagenomic samples, hopefully. So the way we constructed our database is we first did some automated data mining using NCBI taxonomy, which has some host information in there, using the database of virulence factors for fungi, which actually is manually curated, so that was nice, and also using the AID2 database, which is a really nice, huge resource, but unfortunately also relies on automated mining. So it does have some small errors in there, and those labels are not at the level that we wanted them to be. So we had to take all that data and curate it manually on top of that. So we used the Atlas of Clinical Fungi, data from the German Plus Resources Information Network, and a bunch of different individual papers, and we tried to actually confirm all those pathogen labels that we got from automated data mining. And if we knew that something was suggested to be hosted by humans but not necessarily a human pathogen, then we didn't use it in the follow-up. And finally, we used some reusable R scripts to also link that to GeneBank genomes, so you'd have pathogenicity labels and genomes and everything like that, so you can directly download that and use it in your workflows. This ends up in a big database. A lot of that is actually taken from AID2, but we have like 10% of those almost 1,500 labels. For about 10% of them, we actually already have genomes that are sequenced. And a bit less than 1,000 of those are manually confirmed pathogens, with some host groups assigned to it, with some diversity on the taxonomic level. And you can see that we can add some new data to H2 and also confirm some of the putative human pathogens that were included there. And for some of them, it doesn't look like that. The database is available here. You can access it, and you can also take the code that we use for everything, and see our paper, of course. The scripts that we used can be, well, used also to update the database. We did it last time in January, but we're gonna do it again this year still. Then, where we take that database, we can actually start doing the deep learning or the sequence alignment part. So we divided our data in the positive class and the negative class for fungi, so human pathogens and non-human pathogens. And we divide them so that the training, the validation, and the test set have completely different species in them, so we can model this novel species scenario. We simulate Illumina reads for all of those, and we pass them to ResNet, so a residual deep neural network that we have developed previously. And you can install the framework that we used developed in our lab to try to reproduce that results or do inference. So we take those simulated reads, it's up to 250 million actually, and train the network. And it turns out to work fast. That's the most important part. So it's way faster than blasting. And why we use blast, not something even faster? Well, blast is the estimated upper bound of how far we can go with homology. So we want to see what's a kind of gold standard in terms of accuracy. And you can see that both blast and the ResNets work pretty well if you have whole genomes or single species samples, but on the read level or read pair level, it doesn't work that well for any of those. I mean, it could be useful in some situations, but it's way worse than what we had for bacteria and viruses. We also ran a temporal benchmark where it looks a bit better, so you can see that it kind of depends on what species you're looking at, but still those read level predictions are only useful in some situations and more like a stepping stone for having our networks generalized to many different sequence lengths, and also to create learned representations of all the genomes in our database. So what we did here is we took the activations of our neural network from the second to last layer, and we averaged them over all the reads coming from the same genome. And doing that, you end up having a nice representation of your whole genome that's actually learned. So you should have some relationship with the pathogenicity potential that we're trying to model. And then you can try to plot that representation in 2D with UMAP, and you end up with something like that. So we have some clusters which are not perfect, not beautiful, but there are some things going on. And actually the interesting thing is that this is related to, but not reducible to fungal taxonomy. So you have some phyla that cluster together, and you have some classes, this is at the class level, that actually end up in different classes. So there's more going on here that would probably have to be analyzed even more. Now, I mentioned that we were a bit disappointed with the read level predictions, but that's just because it's really difficult to differentiate between fungi that infect humans and fungi that infect other organisms, at least when you have only 250 BP. That was not that surprising. But we would like to have this work on metagenomic data, and let's assume here, for the sake of the argument, that we have a human/clinical sample. And in that sample, we would like to do readwise predictions still, because assembly is difficult and because we want to have very fast predictions. This is important in a like potential outbreak scenario. But we can assume that we're only going to have human-hosted bacteria in there, but we're going to have both human and non-human viruses, like bacteriophages that infect the bacteria that infect humans. But we're only going to have the human-hosted fungi. We can, fortunately, ignore this one non-human pathogen class for now. If you took that from a human, it's probably infecting humans. There is another problem here. So bacteriophage sequences tend to be quite similar to bacterial sequences of, well, to the sequences of the host that the bacteriophages infect. So that's another decision boundary that's hard to learn that we'd like to avoid. So we'd like to put all the negative classes together. So we're going to have a joint negative class of things that are harmless to humans. We're going to have the human pathogen bacteria class, the human pathogen virus class, and the human pathogen fungal class. And this actually turns out to work pretty well. Also on the read level, this is actually performance for read pairs, but you can see that we're way better than BLAST, which was the expected outcome and also faster by that way, in all those different classes. And it's interesting to see that it also works for real, not simulated, sequencing runs. And somehow worryingly, it's way better than STAT. STAT is a k-mer-based approach that's used by the NCBI SRA database. So if you put your sequencing runs there, you can have like a taxonomic assignment of your reads. And unfortunately, it seems that STAT, at least in this cherry-picked case, really underperforms when it's, well, it finds an unknown fungus. And underperforms means it doesn't even classify those reads as fungal reads, which we can do. So, summing, yeah, that was disappointing. I saw some surprised faces. So yeah, summing this up. We have a collection of fungal genomes that was useful for our research project, but we hope it could be also useful for many others. Those are basically genomes with labels and sources for those labels. That's, well, we hope could be used also in proteomics, functional genomics, whatever you do. We have predictions for novel agents, novel viruses, novel bacteria, novel fungi that are better than standard sequence homology. And this works on genomes, reads, at least under some provisions, and potentially also synthetic oligonucleotides. The goal here is to fast or even sometimes real-time detection of novel threats, and for that I would have to refer you to other papers from our group. And yeah, the database is accessible under this QR code, and you can also install the code, the package that we use to train and also do inference with either Conda on Docker. It's pretty accessible, so if you don't want to train your own models but just use ours, it should work too. With that, I'd like to acknowledge our cool group and Katharina Baum, who also helped us a lot, and I'm happy to take any questions.
Plenty of time for questions.
Hi, Jacob. Thanks for the talk. I mean, I'm really happy to see that there is some pathogenic fungi here. I was wondering, so there can be differences between strains of the same species sometimes. Even few mutations can generate like avirulent strains. So I was wondering how well does this DeepPack tool work in this situation for this classification?
Yeah, you're right. That's a very crucial thing. So, I mean, the model will reflect your assumptions in the data. So, the tool will basically reflect whatever you decide. Mm-hmm. What we decide usually is we take some taxonomic level, and in this case, it's a species, and we try to, well, in a reductive way, put admittedly, assign a label to it. So usually try to be on the sensitive side of things. So if that species is reported to cause disease in humans, we count it as pathogenic. And we, well,
so
then we would
per
assign
species.
a strain that didn't cause a disease as pathogenic. But I would say this is important, especially since you have a lot of immunocompromised people. So, I mean, you can have a strain that looks like it doesn't cause a disease, but it could. So we're trying to predict potential rather than pathogenicity itself.
Okay, cool. Thanks.
Any more questions?
Hi, nice talk. I was wondering how well it would fare your package against genomes of hybrids. As we know, some of the pathogenic candidates may be a hybrid, so I was wondering, would it perform as well as with single normal candidates or other pathogenic fungi?
We have not checked hybrids. We actually excluded them from the database because that was getting difficult. I would assume that it would work. So the way we handle genomes is actually divide the genome into overlapping sliding windows, and we predict for every of those windows and then average the predictions. So in the case of a hybrid, you would probably basically see predictions resembling one part of the genome and the other part of the genome, and kind of then depends on which pieces got hybridized. So yeah, we had a project previously that showed that those local predictions should be somehow associated also with virulence factors, at least in bacteria. So I'd hope, although we didn't check that for fungi, maybe that could also be somehow relevant. So hopefully, if you get virulence factors from one part of the species, it's going to matter. But we didn't check that, so yeah.
And a follow-up question. So you mentioned that for the first problematic was selected only one model of pathogenicity interaction. For those cases that you may find horizontal gene transfer in a specific genome, and that horizontal gene transfer may be linked with a pathogenicity label, would be able to be detected as such, or because you're limiting to only one type of interaction, let's say human fungi, but perhaps that fungi has suffered an event of horizontal gene transfer with a fragment of a gene of a bacteria that has, for example, a pathogenetic label. So how would it handle those kind of situations?
So yeah, it's an interesting question. What would happen if you get a transfer from bacteria? We didn't think of that. In general, yeah. So in the multi-class model actually assigns probabilities to all those classes. So if you really had a piece of DNA that looks very bacterial, so to say, that piece of DNA would probably be classified still as a bacterium, or at least it would be confused whether it's a bacterium or a fungus, like depending on some sequence features. But those are local potentials, and those should get kind of averaged out when you look at a longer sequence.
Okay, thank you for your reply.
Thank you so much.
Any more from the audience? We have one from the chat, so I'm going to read it. Maybe I missed it, but how would a non-fungal eukaryotic pathogen will be dealt with in your system?
A non-fungal eukaryote. Yes, that's something we did not model at all. That's a really great question. Unfortunately, it's even harder to get good data for that. So, I mean, of course, there are good data, but you need a lot of species taxonomic diversity to train the deep learning models. So, for now, we ignored that, which is a major limitation, I must agree. I would assume that it would probably be classified as fungal, at least some parts of it, but it would have to be checked. So, yeah, it could depend on the model. It could either treat fungi as, representatives of all eukaryotes, and then it would get into fungi, or it could be so far out of the distribution of the sequences that we modeled that you would get very unpredictable results all over the place.
There's no reply in the chat, so should be fine. And I was curious when you said non-human means that you're including every fungal infecting everything, plants or animals, anything else. But should be perhaps a big difference, I don't know.
What do you
Fungal infecting plants, for instance, are really different from the ones infecting animals? Or it's better just to put all them in a single pack?
Yeah, okay, that's what I mean. Yeah, also you don't have that many fungi that infect animals and don't infect humans. You do have some, but it would be probably difficult to treat that as a separate class. And since we're trying to do like a very simplistic classifier that you can then use also quickly in your pathogen detection workflow, we are kind of interested also in a yes or no kind of question. But it's very human-centered, I agree.
Is there any more from the audience? I always am curious when you say manually curation, who's going to do that in some future? So it's the same question that was in the first call. How is this the maintenance of this database, this software planet? I mean, you need data, you have to query data. This is a really quick evolving field. How is you improving the database or getting pace of everything that is new species, new data? Is any plan for maintenance, long-term maintenance of this database?
The plan for long-term maintenance? Yeah, I mean, that will depend on the user base mainly, and like what's the need from, if we get users, I hope so, invite you cordially. That will depend on that. So of course we plan to update it, but from our perspective, that would be probably maybe once, twice a year or something like that. But if it's needed, please talk to us, and we can do it maybe faster or collaborate to get more people on board.
Well, that's a typical issue, maintaining things once they're done.
Yeah.
I don't see much more questions, no more questions, so we can just close it here. I'm afraid that for the ones that were late, that we missed the last speaker. I'm sorry for that, but apparently he has had a problem to come and he was not going to be here. I will thank Jacob and Alexander for the wonderful talks, and I will let you go to lunch.